Okay, in this audio lecture, we are going to look at cell structures that are found in microbes. So we'll be talking about prokaryotes, eukaryotes, and viruses. So information that's covered in this audio lecture are in chapters 4, 5, and 6. So chapter 4 is prokaryotes, chapter 5 is eukaryotes, and chapter 6 is viruses. So when we look at microbes, we really can look at them through an evolutionary timeline, with prokaryotes being the first to appear well over 3 billion years ago, where eukaryotes date to about 2 billion years ago. And if we look at eukaryotic microbes that are multicellular, like fungi, they would be even newer. And again, if we think about this evolutionary and compare this to us humans, we've only been on this planet for a little blip of that. And so, again, looking at cell structures, prokaryotes are going to have more basic cell structures, so they lack some of that specialization that we see with organelles. So again, one of the big hallmarks of a prokaryote is that they lack organelles. They have cell structures. With eukaryotes having specialized cell structures known as organelles. So this figure just shows us some of the different types of microbes. Our microbes can be broken into two different categories, cellular and acellular. For our cellular microbes, those would be either prokaryotes or eukaryotes. Examples of prokaryotic microbes would be bacteria and archaea. Archaea are more primitive. They tend to live in really extreme environments like lava fields and thermal vents. And so from a medical standpoint, archaea don't seem to have much of a role because we're not good hosts for them. There's some newer studies out looking at archaea and whether they play a role in teeth and cavities and such. But, for most textbooks that have any kind of medical human aspect of microbiology, there's like that one line, and that's about it. So the focus of this semester for us will be bacteria. So if I say prokaryote, you can kind of default and think bacteria. Just know that there are exceptions. Those archaea are really unique and special. If we look at our prokaryote, it does have cell structures. It has a cell membrane, a cell wall, ribosomes, chromosomes, and flagella. So they don't all have these structures. And we'll talk a little bit about what structures all cells do have in a little bit. But those are just some of the cell structures they have. But you notice that in that cytoplasm, they're not going to have the organelles that we see in the eukaryote. So if we look at the eukaryotic figure here, you can see that we have a nucleus where the genetic material is going to be enclosed. We have mitochondria that are going to be the powerhouse of the cell, where ATP is synthesized. And we still have a cell membrane, we have the ribosomes present, and we could potentially have flagella present. Examples of eukaryotic microbes would be fungi, protozoans, and hemolyphs, or parasitic worms. Now, when we look at our acellular microbes, these would be viruses, and those are further broken into viruses that would infect, animals, plants, or we can also have bacteriophage. Bacteriophage are unique. They infect bacteria, so they're viruses that infect bacteria. And you can see that bacteriophage in that figure that kind of looks like a space shuttle with this with these little arms. Those are actually pins that actually will anchor it to the surface of the bacteria, and then that kind of neck on it, that's the sheath. That's actually very much like a hypodermic needle, in which the nucleic acid that's in the capsid will get injected into the bacteria. Once that nucleic acid is inside, the bacteriophage will replicate, make copies of the nucleic acid, make copies of the proteins that are needed for the capsid, any kind of enzymes needed, and then resynthesize new bacteriophage. Also in that figure, we can see we have a human virus. So this is the AIDS HIV virus, and so you can see how it has an envelope that encloses it. And then we also have that capsid and nucleic acid. Just to note, not all viruses have an envelope. So some viruses are non-enveloped or naked viruses. So viruses come in different types. And then also the nucleic acid can differ. So we can have either DNA or RNA, but viruses aren't going to have both. So when we look at prokaryotes versus eukaryotes, one of the things you'll notice is that the size is different. If you were to look at them underneath the microscope, if you think back to anatomy and physiology and you looked at eukaryotic cells, you could see them at the 10x, and you might have gone up to 40x to get a little bit more information, but you never had to go to 100x. Now, if you are doing a face-to-face lab or micro lab, or if you've seen images of microbes underneath prokaryotes underneath the microscope, you have to go to a higher power. So you actually always have to use the 100x and that with oil because they're so much smaller. And that's in part because they don't have these specialized structures. So all the material has to be able to diffuse and move throughout the whole entire cell. So that really limits its surface to volume ratio. Where with eukaryotic cells, because they have those specialized structures, those organelles, and they have a more developed transport system, they can transport vesicles from one location to another. So I always think of like ATP. If I'm a prokaryotic cell, I'm only able to make ATP on my cell surface. They actually make it on their cell membrane. And so I need to make sure all parts of my cell are close enough to the cell membrane that I can get ATP to it, so that we can catalyze reactions and such. With a eukaryotic cell, we just need to make sure mitochondria is nearby, so we can space the mitochondria out, and that allows it to be much larger. So if we were in a classroom, I'd bring a bunch of tennis balls, which would represent ATP, and we would say you have to be able to gently toss that tennis ball in all areas. And one of the things you would notice is if you guys were acting like the cell membrane, you have to move close together in order for those tennis balls to get to all locations. But if you were a eukaryotic cell and you were now acting as mitochondria, you don't have to be as close, and you can actually move further out. You can social distance inside that eukaryotic cell that we're making, and so that it just allows the cell to be larger. Okay. So in this audio lecture, we'll look at different cell structures, and one of the things you want to do is to think about, is this a cell structure that's found in all cells, most cells, or some cells? So I would highly recommend, normally I project these up onto the board, and I write in the different cell structures that fall into this as we're going along the audio lecture, but definitely something you can do in your notes. There are 4 cell structures that are found in all cells, so you should definitely know that. So all cells are going to have a cell membrane. You can't be a cell without a cell membrane. That's your boundary. That's what creates the outside versus inside. We also have to have cytoplasm. Cytoplasm is going to be the fluid which allows material to move around. We also need to have ribosomes. So ribosomes are going to be the structure that allows us to take nucleic acid, RNA, and make proteins. So cells are going to need to be able to make enzymes, proteins, and so we need to have ribosomes present. And then lastly, we need to have that instruction manual. We need to have genetic material in order to code for genes that then can be turned into proteins. Okay, so again, those would be the 4 cell structures all cells have, and so everybody should know that. And then as we go through the lecture, you'll hear about some of the most and some categories. Keep in mind that depending upon whether you're talking about prokaryotes or eukaryotes, some of these structures can shift from that most to some category. Okay, so the way we're going to do this tour is we're going to start on the outside of a prokaryotic cell and then move inside. So we're going to get on our magic school bus and pretend we're viewing the cell from outside, and we're going to look at these external appendages and structures that are present, and then we'll move into the cell envelope, and this is made up of several structures in a prokaryotic cell. and then we'll move internal. And as we move through these prokaryotic structures, we'll compare them to the eukaryotic structures, and then we'll wrap up with any kind of like specialized features, and a lot of those are internal structures. So this is a figure that just shows you all the possible cell structures that could be on a prokaryotic cell. So this is a nice figure to study from because it names the cell structure, it gives you an example of what it looks like, and then also provides some information about it. So again, that's really nice. And you also notice the shading is a little different. So the darker shading are the ones that are found in all prokaryotic cells, and they're actually found in all cells, also eukaryotic cells. And then the lighter shading indicates that they're either more, most, or some. Okay, so we'll go through and we'll talk about these. So I won't spend any more time on this figure, but again, it's a very nice figure to go back to and reference as you're listening to the audio lecture, but also as you're sitting for the exam. And to compare, we have our eukaryotic cell. You can see there's a lot more going on in here. Additional structures or organelles that are present. Honestly, I don't like this figure as much because it doesn't give you as much detail, but again, you can look at some of the cell structures that are common between prokaryotes and eukaryotes, and again, we'll talk about that too. So looking at our external structures, especially our appendages, these are going to be cell extensions, so they're going to extend out of this the body of the cell. So they are very common, but they're not necessarily present in all species. And some species would only have one of them and not all of them. Okay. Some of these structures provide motility or movement, and some of these motility structures are found in both prokaryotes and eukaryotes, and then some are unique to prokaryotes and some are unique to eukaryotes. So Flagellar is a mobility structure that is found in both prokaryotes and eukaryotes. They're a little bit different, and there's actually new research that was published over the summer about the eukaryotic flagella that was really cool, but we'll focus a little more on the prokaryote because you're probably not as familiar with that. You probably would have talked about eukaryotes in like a cell bio class or intro course that you might have taken. And then another structure we have are axial filaments. So these are more or less embedded flagella that are found in some prokaryotic cells, a special group called spirocytes. And so they actually wiggle and everything. So that's a really cool structure that's unique to prokaryotes. And then we have cilia, which are found in eukaryotic cells. So cilia are not found in prokaryotic. And so you're probably familiar with cilia because maybe you looked at pond water underneath the microscope and you would have seen these parameciums that can move around, and that's usually, they have little cilia, little hair-like projections on them. We also have pili and fimbriae. Those are appendages, external structures that allow for attachment and mating. So fimbriae would be the attachment, and pili attaches, but it also creates like almost like a straw-like structure between two different prokaryotes. So they actually allow for genetic exchange. So not the same kind of mating as some eukaryotic organisms, but again, it's the idea of this exchange of genetic information through the pili. So just looking at the basic structure of a bacterial flagella, it works like almost like an electric motor. So it's got this basal body, and it's going to be very high ATP demanding, and it has a filament coming out of it, and so it actually will rotate very much like a propeller. So it drives, just like a propeller drives a boat, through its environment. Okay, you don't have to memorize the individual structures, just remember that it has a single filament, and that becomes important when we look at the eukaryotic flagella. With prokaryotes, there's lots of different arrangements that can happen. So we can have where we only have one flagella, and that would be monotrichous. Trichous stands for hair, so one hair. Amphitrichous, this is where we have two, and that's seen in panel C. And so on both ends, ambi, think of amphibians, land and water, and so on both ends of the cell body of the prokaryote. Lophotrichous, which we see in panel B, lopho meaning tough, so like it's like a tuft of hair, a cluster of hair, so it's found on one end, and there's multiple flagella. And then we have peritrichous, and peri meaning throughout, and this is where the flagella are found throughout. Okay. So the function of the flagella is motility, movement, and so this can be through chemotaxis, so it can be drawn towards a chemical signal or away. So if it's drawn towards, it would be a positive chemotaxis. If it's drawn away, it could be negative. In some cases, it's actually the movement of the flagella is regulated by phototaxis, photo being light. So there are bacteria that are scared of the dark, so they will actually move to the light. Usually this is because they undergo like photosynthesis too. At the same time, there's bacteria that are scared of the light. They like the dark, and so they would move away from the light. So we tend to think with eukaryotic cells that have flagella, they move in a very driven path. Prokaryotes don't. They actually move in what's known as a run-tumble pattern. So here in this figure, we can see how we have these tumble moments where the bacteria basically just tumble around if you were looking at them underneath the microscope. And then they have run moments where all the flagella are working, they're not working together, but they randomly move in a way in which allows for this run pattern. And then that moment that they're working together just goes away and they start tumbling again. So what ends up happening over these tumble run patterns is that they end up moving towards something or away from something. If we were, in a class, I would do my little example, my arms flailing where I'm in a tumble and I'm spinning around, and then all of a sudden I would like start moving somewhat coordinated towards something and then start tumbling again. But you can imagine how if we were watching a bacteria under this, we could see these patterns. And again, if it's chemotaxis, it's going to be moving either towards a higher concentration or away from a higher concentration, where if there's no attractant or repellent, that movement would just be in a uncoordinated direction. Now, looking at the eukaryotic flagella, one of the things you'll notice is it's much larger. There's more filaments to it. So it actually has this 9 × 2 + 2 pattern. And so if you look down on the panel to the right, you can see how we have these groups of 2, and there's actually 9 of them towards the outside, so 18 microtubules. And then in the middle we have additional 2, so 20 microtubules, filaments coming together to create the flagella. And so because of that, its movement's a little bit different. So it actually has more of a back and forth movement, though research over the summer came out that there is a twisting motion to it, so it's not as simple as like a waving back and forth. So I used to be like, oh, just go back and forth, but more complex than that. ATP is really important in this movement. So again, if you've taken like a cell bio class, you would have talked about motor proteins, and that allows for binding to the ATP and actually shifts and slides these tubules back and forth. A little more information than you really need to know. Again, it's just that if we were to compare and contrast the prokaryote and the eukaryotic flagella, the prokaryotic flagella only has 1 filament, where with the eukaryotic there's 20 filaments. They both have the same function of motility. Now when we look at cilia, structurally they're very similar to flagella, except they're shorter, and there tends to be more of them. So in many organisms, protozoans, you would see cilia throughout the surface of it, just like that paramecium. There are some that actually use the cilia when it beats, when it moves, it actually causes food to come in, so they can be used for feeding and filtering, not just motility. Okay, and again, a structure that's unique to eukaryotes. We do not see cilia on a prokaryotic. Now looking at the axial filaments, these are our spirochetes, and so again, as you can see in that panel B, all those little circles are little flagella. So same flagella protein, but they're actually embedded into this periplasmic space. And so what happens is you get contraction and relaxation and contraction and relaxation, going back and forth on the different sides of the bacteria. And so you end up with this actually corkscrew movement, and it actually allows the bacteria to move through pretty viscous material and into tissues. So if you do research on spirochetes, these are some pretty nasty bacteria, like syphilis, leptospirilla, and Borrelia. You might recognize Borrelia because that's the agent that causes Lyme's disease. And so one of the issues with Lyme's is the symptoms can be so varied because it all depends on where the bacteria ends up corkscrewing into. So it can go into joints, it could go into brain, it actually can corkscrew into red blood cells and hide in the red blood cells. So again, spirochetes, because of these axial filaments, are pretty nasty. So looking at fimbriae and pili, so you can see on our figure here, we have fimbriae here, and that bacteria has kind of a fuzzing around it, and so if we were actually, and the next slide shows it's better, you can actually see these individual little structures. So very much like cilia, in that they're these like short hair-like projections, but these allow for attachment. In some cases motility, but again, more an attachment. So I think of them as like little Velcros. So they will Velcro themselves to host cells. And again, we'll see this a little more in the next figure. For pili, you can see those little projections coming off of the bacteria attaching to other ones. These actually are forming like a straw. And so genetic material can actually be moved through that hollow structure from one bacteria to another. So if I'm a bacteria and I have a plasmid that codes for antibiotic resistance, that's good, right? Well, I could share it with my other bacteria friend, and so I would make this pili so we could connect. So in this in this set of figures in panel A, what they've done is immunofluorescence. So they've taken antibodies that are labeled that the antibodies recognize the fimbriae proteins, and they're labeled with the dye that shows up as yellow. So we can see how we've got these like little yellow projection hair-like structures indicating that we have fimbriae on the bacteria. In the bottom panel, we have an SEM image, scanning electron micrograph, and so we can see how these E. coli cells are actually attaching to the intestinal microvilli, and so they're actually using these fimbriae in order to do that. So we want to keep in mind that bacteria cells are very dynamic, and so an E. coli can produce these fimbriae, but then stop producing them. And so this is one of the ways that, if E. coli senses that there's competition or something going awry, so maybe there's salmonella that's moved in because you ate a bad hamburger. So your beneficial E. coli will actually stop producing the fimbriae because there's an enemy out there. There's something that might attack it. And when it stops producing the fimbriae, it detaches from your intestinal microvilli. It also sends signals, it produces hormones that causes your intestines to move faster, and so that the E. coli can get out of there. So it doesn't have to compete for resources with the salmonella. It'll probably find a new host at some point, right? So again, just keep in mind that some of these structures are very dynamic. It's not does it have it or does it not. It depends upon the environment it's in, whether it's going to express and have fimbriae or not, whether it'll have a flagella or not. So again, they're very dynamic in that sense. So another external structure is the glycocalyx. The glycocalyx is a polysaccharide protein coating, and so I think it's like a sugar layer, and it actually helps to protect the cell, and in some cases it can actually adhere to an environment. Glycocalyxes come in two different varieties. There's what's known as the slime layer. So the slime layer is going to have this glycocalyx being loosely attached. So it actually gives it a slimy appearance. So if you look in panel B, it's actually the one on the right, the one that has that kind of like wet appearance to it. So that would have a slime layer. And so this actually helps the bacteria from losing water and nutrients. But these slime layers can actually interact with each other and interlock and create a biofilm. So in that sense, glycocalyxes are really important medically because we know biofilms are important. The other type of glycocalyx are capsules. The glycocalyx is tighter bonded to it. So I think of this as like the the candy coating on an M&M. So it's all made up of sugar, and so much more tighter. And so you can see on the bottom panel, this is what's on the left, and so that has a kind of dry appearance to it. So the capsule also helps to prevent loss of nutrients and water, but it also makes it very difficult for immune cells to actually attach to it because those sugars are some of the same sugars you have, because eukaryotic cells also have glycocalyx. So an immune cell will kind of attach to and be like, I don't know, that's sugar. That's a sugar my own cells have. No, don't respond. Okay, so it's a way to kind of trick the immune system, and it also makes it for adherence to be a little bit tougher. So as I just mentioned, the functions of the glycocalyx, it actually helps to prevent bacteria from being, protects the bacteria against phagocytes. Phagocytes are what are trying to engulf their immune cells. Okay, it's also important in biofilms. So if you think about, I don't know when the last time you brushed your teeth were, but you get that film on your teeth. Those are actually biofilms starting, and so those are bacteria that have slime layers that they're producing. So now we're going to move into the cell envelope, and this is that transition point between the outside and inside cell. And so with eukaryotes, for the most part, for like our protozoans, they're just going to have a cell membrane. For our bacteria, they're going to have the cell envelope can be made up of several layers. In some cases, some bacteria only have a cell membrane. Most bacteria also have a cell wall, so they would have 2 layers in their cell envelope. Some bacteria also have an outer membrane, so they would have 3 layers. And so depending upon how many layers bacteria has in its cell envelope, lets us identify what type of bacteria it is. So one of the things we'll be talking about in the lab is gram staining, and so some bacteria are gram positive, some are gram negative, and some are none of the above. So right now we're going to focus on gram positive and gram negative and forget the outliers for a moment. So if you are gram positive, you're going to be a bacteria that has 2 layers. You're going to have a cell wall and a cell membrane or cytoplasmic membrane. And then if you're gram negative, you're going to have 3 layers, so you also have that outer membrane. So looking at an SEM, scanning electron micrograph, that they've added pseudo-coloring. So what they've done is actually made it easier for you to see the different layers. On the left-hand side, we have a gram-positive bacteria, and you'll notice the cell wall, which is in brown, is very thick. That is made up of peptidoglycan, or PG. So this peptidoglycan is the subunit that makes up the cell wall. Okay, in bacteria, in prokaryotes. Keeping in mind that archaea, their cell wall is made up of slightly different material and also their cell membrane. In that yellow, we can see where the cell membrane is. Now, if we compare this to the right, where we have our gram-negative bacteria, You'll notice that that cell wall, that peptidoglycan, is very thin. So it's just like they took the brown mark and made a really thin mark. Just like the cell membrane, that yellow is also very thin. The outer membrane is present, so that's shown in the green. So we don't see that in the gram-positive bacteria. So as I mentioned, the structure of the cell wall, it's going to be made up of peptidoglycan. This allows the cell wall to be rigid, so it provides a support structure similar to like in a plant that has a cell wall, right? So you're probably more familiar with plant cell walls, so they provide that. What's the shape of the cell going to be? And so same thing with prokaryotes, with bacteria. Bacteria come in different shapes, and so that shape is determined by the cell wall. It's interesting to note that if you don't have a cell wall, you can be lots of different shapes. So this is just showing you kind of how the peptidoglycan actually cross-link together. So if you think about chemistry, you can have different orientations of the structures. So referred to as chairs. So we can have these different orientations, and then they end up getting cross-linked to each other, then also cross-linked in between these different chains. Okay, so you can see those cross-links there. That becomes important because when we look at antibiotics, some antibiotics will actually break these cross-links and prevent the repair that happens. So the cell wall is going to be really important so that the bacteria can maintain its shape, maintain osmosis, so water pressure inside and outside of the cell. And so any kind of rupture, damage to the cell wall will affect the ability for the cell to maintain homeostasis. And so again, there's antibiotics that are actually going to disrupt the cell wall, and so it actually will lead to the cell lysing and dying. So your penicillins and cephalosporins are antibiotics, and we'll talk more about those later on this semester. Okay, so as I mentioned, again, gram-positive has that thick cell wall, where with the gram-negative there's that thin cell wall, but then there's also that outer membrane. And in case you didn't like the scanning electron micrograph image, here's a cartoon drawing figure showing gram-positive versus gram-negative. Okay. So we want to keep in mind with the gram-negative, it has that outer membrane. That outer membrane is going to contain lipopolysaccharide, or LPS. And so that becomes important, and we'll talk about that in just a moment. Another feature with the outer membrane, because this outer layer, it actually makes it hard for molecules to get in, such as antibiotics. So a gram-negative bacteria is going to be less susceptible to the antibiotics, so you may have to do a higher dose. Also, their cell wall is thinner, so if it's a cell wall in antibiotic, it's not going to work as effectively against an outer membrane or a gram-negative bacteria because of the outer membrane. So as I just mentioned, the outer membrane is going to have lipid polysaccharide, or LPS. LPS has 3 domains. There's a lipid A, or an endotoxin present. There's also the core polysaccharide, and there's the O antigen. Now the O antigen, immune cells can see this, so that seems like a good thing. So it actually causes immune cells to target gram-negative cells, but in the process of doing this, it actually releases lipid A. Lipid A, endotoxin, a toxin within. So when the gram-negative bacteria dies, it releases lipid A. Lipid A can be incorporated into any cell that has lipids, and if you think about that for a second, that would be any cell. So if we have a gram-negative bacteria living in us and we've taken an antibiotic that's going to kill it, or maybe our immune system has come in, potentially we're releasing lipid A, and that can get incorporated into our own cells, and then it can cause those cells to die. We're multicellular, so as long as there's not large amounts of endotoxin, we're able to deal and repair those organs. But obviously, if there's a large amount of gram-negative bacteria, large amounts of lipid A, that damage could become so severe, and we can end up going septic and potentially dying. Okay, so we'll talk more about this later on in the semester when we talk about some of the pathogenicity of bacteria. So I just briefly want to talk about the process of gram staining. We'll talk about this more in lab. And so it's a 4-step process, which we start with crystal violet, and crystal violet is going to bind to the cell wall. So keep in mind, gram-positives have a thick cell wall, so there's lots of binding sites. The next step is grams iodine. So that's actually going to bind to the crystal violet and make it so it's larger, so it's harder for the crystal violet to come out from the cell wall, so it actually gets trapped. Then we have alcohol that is going to decolorize, remove any excess crystal violet, and actually punches holes into the outer membrane, so it's easier for the dyes, any further staining to get into the cell. So then the last stain is saffron, which is red, and so because the gram-positives still have that crystal violet iodine complexes present, the red's not going to show up, right? With our gram-negative, because they didn't have a thick cell wall, they also had the outer membrane that was blocking the crystal violet from coming in, and we just punched holes in the outer membrane, that red stain, that saffron, is actually going to stain the cell. So a gram-positive cell is going to look violet or purple, where a gram-negative cell will look red or pink. Okay. Again, you don't have to memorize these steps when it comes to the lecture piece, because we will be talking about this in the lab piece, and you'll be assessed on it and tested on it there. But you should know, when we're talking about a gram-positive bacteria in the lab, it would look purple. A gram-negative bacteria would look red if we did a gram stain. So as I mentioned, there's also other possibilities besides gram-positive and gram-negative. So there's these non-typical cell walls, and also some don't even have cell walls. So Mycoplasma is one that lacks cell wall. So we'll look at that in just a second. As I mentioned, Archaea, they have really these unusual and chemically distinct cell walls. And then Microbacterium, they have unique lipids that are present. So the traditional gram staining won't work for them. So for Mycoplasma, this is what it potentially would look like if you looked at it underneath the microscope. This is a scanning electron micrograph, but if you were to look at it underneath a light microscope, what you would notice is the shapes are going to be all different. So some of them could be coccyes or spears, and other would be filamentous, like long, thread-like looking. Okay, so Mycoplasma pneumonia is probably one of the more commonly known important medical species. So then we also have the acid-fast bacteria. So this would be the Mycobacterium, and again, because they have unique lipids present, they don't react to the gram staining the same. So they might look gram-positive, but they're not truly gram-positive, and so you have to do a different staining. So their acid-fast has an acid stain that actually helps to break down some of that lipid so that you can actually stain them color. Okay, so tuberculosis, leprosy, and some of the opportunistic wound infections that happen would be medically relevant pathogens or acid-fast bacteria. So clinically, if you had a patient that had an infection, one of the first, and you sent out culture, they would do a gram stain, but they'd also do an acid-fast stain. Now, when we look at the eukaryotic cell wall, it's also going to be very rigid and provide support and shape, but it's going to be chemically distinct and different. So for thinking about, we as eukaryotes, humans, don't have cell walls. Plant would be covered in botany. So thinking about the eukaryotic cell walls, we need to be concerned with in microbiology, fungi. So fungi the subunits would either be chitin or cellulose. that makes it up. And then we also have algae, and algae would have cellulose or pectin. So the way I think about it is prokaryotes are going to have peptidoglycan, fungi are going to have chitin, and then algae are going to have pectin. Okay, so those are the unique subunits to each of those groups. So if you've ever made a jam, you might have used pectin, so isolated from algae, to thicken up the jam, right? So cell walls are going to have that kind of consistency where they're going to be thicker. So next, looking at the cell membrane and its structure. So in the most part, this is going to be very thin. So that's why, we have to do the scanning electron micrograph and put the colorization so you could see it. It's going to be made up of phospholipids and proteins, and just be aware that there are always exceptions. So archaea and Mycoplasma, they actually have sterols that are incorporated into their cell membrane. But the cell membrane is going to provide the function of creating that outside inside. Also with function, we're also going to have regulation of transport. So we've got that selectively permeable membrane. So it, again, is selecting what can go into the cell and what can leave the cell. Also the process of secretion occurs through the cell membrane. And then something that's unique to prokaryotes is the ability to produce energy. So they can undergo energy reactions, and they synthesize ATP on their cell membranes. So again, that's something unique to prokaryotes. With eukaryotes, we have mitochondria. Full disclosure, there are some eukaryotic protozoans that actually do not have mitochondria, and so they still are able to produce ATP over their membranes. But in general, this ability to produce energy is unique to prokaryotes. Okay, so this will conclude part 1 of this lecture. I will be capturing a second part that will look at the internal structures in cells.